Hello, my name is Pratyush Chera, and today I'll be presenting my journal discussion on label-free microarray imaging for direct detection of DNA hybridization and single nucleotide mismatches. Now, a DNA microarray is effectively a glass plate that has individual little cells, and on each little cell there is single-stranded synthetic DNA that has been specifically pre-designed to bind to DNA sequences on introduced samples. Now, what normally occurs is that you take an unknown sample, fluorescently tag that sample, then apply those samples to the DNA microarray. These samples are split into single strands, and they will then naturally hybridize to complementary strands on the synthetic DNA. When this occurs, you can then apply a wash in order to knock off the strands that did not properly bind to a synthetic, and then use spectroscopy to fluoresce the strands that did successfully stay on the plate, allowing you to visualize the gene sequence that you're analyzing. Now, one of the primary use cases for this technology is in the detection of single nucleotide polymorphisms. Now, a single nucleotide polymorphism is effectively a single base pair mismatch, which can have severe impacts on the proteins that are being generated by the gene, though not always. And it's very important to detect, as many diseases can either be caused or their development changed by specific nucleotide mismatches. However, when attempting to use the microarray technology, you run into a small problem. The standard microarray depends on the idea that your synthetic sequence will bind to a matching sequence in the sample and won't match any other way. In reality, due to the fact that only a single base pair is mismatched, you can have multiple configurations where a bond is successfully formed without it being a perfect match, and you otherwise wouldn't know that. So it's very hard to do this with standard fluorescence. So the solution that this paper proposes is to use a label-free microarray using spectral reflectance imaging biosensors, which is quite a mouthful, but I'll try and explain it to the best of my ability on the next slide. So this method attempts to, instead of looking for a fluorescent signal, measure the optical thickness of the sample and attempts to gather quantitative information of bondings between the analytes and the probes. It does this by measuring the degeneration of the DNA samples over time by the introduction of specific ions which cause the samples to denature and break apart, lowering their thickness and their density, and thus lowering their reflectance. Very specifically, this is measured over a period of time instead of at a single moment, and thus you can then fit a decay function to these rates. Using these decay functions, you can then separate between perfect matches, partial matches, and failed matches, and double mismatches. And thus you can separate the hybridizations far more finely than you could previously, and thus detect the single nucleotide pair mismatches. Now, the conclusion in the paper is that this system is capable of detecting hybridization reactions using a relatively simple configuration, and that the hybridization of the DNA is clearly detected by measuring the mass density of the spots prior to and after hybridization, and thus measuring the rate of denaturation. The future goals outlined by the paper is to study the denature rates due to temperature changes, and they state that they envision the system will be a prominent tool for DNA studies with a throughput of 104, which demands high data accuracy and simplicity. I do have a certain problem with their results section and their methodology overall, in that, at least within the context of this paper and within the data that they cite, they do not directly compare their separation to a standard fluoroscopy separation, if it is at all possible to do so. And they also fail to convince me that a host of other factors could not have affected these denaturation rates, including temperature, but also specific concentrations in the ions, minor variations in flow rates, and a host of other factors that could significantly impact their rates of decay. And while they do indicate this somewhat in the paper by having very large bars of separation and allowable error, I don't feel it is sufficient to define this technology as the future, at least within the contents of this journal.